AIDS, the most devastating biological catastrophe the world has ever known. This disease will kill more people this year than all other viral diseases combined. According to Dr. Robert Strecker, AIDS is a man-made, genetically engineered virus that was either accidentally or deliberately introduced into the world's population. AIDS is not a homosexual disease. AIDS is not a venereal disease. AIDS did not originate from the green monkey. AIDS is not prevented by the use of condoms, and AIDS is not likely to ever be cured by a vaccine. I realize what you've just heard contradicts most of what you've been told about AIDS. In this program, Dr. Robert Strecker will present documented evidence that refutes the official stand taken by so-called AIDS experts, members of the research community, and the government. And now, let's find out the truth about AIDS.
I'm Dr. Robert Strecker, a practicing internist and gastroenterologist in Los Angeles. I have a special interest in pharmacology, pathology, and now AIDS. I became interested in the AIDS question several years ago in doing a health maintenance proposal, basically an insurance proposal for Security Pacific Bank, a bank here in California. More and more members of the medical and research community, such as Dr. Peter Duesberg of the University of California at Berkeley, Dr. John Seal, a member of the Royal Society of Medicine of London, and Dr. Alan Cantwell, who has recently finished a third book on AIDS, this one on the origin of AIDS, are questioning the validity of the popular view about AIDS, which has failed to scientifically explain the disease. So I've decided it's time that someone tells you the truth about AIDS. So in this program, I will show you how the AIDS virus was actually predicted, requested, produced, deployed, and now threatens the very existence of mankind because it works. To understand why I believe that the AIDS virus came out of a laboratory rather than out of the jungles of Africa, you have to understand several important concepts which I will address in the next few minutes. These concepts actually include an understanding of viruses, bacteria, human cell lines, tissue culture, and manipulation of all of those things in the laboratory. In the first question about the AIDS virus, in addressing what the AIDS virus is, the virus has a morphology, something of this form, which is actually a so-called D-type retrovirus. So what are viruses? Some people say that viruses are the smallest replicating microorganisms. Some people say that viruses are bad news in a sugar coat. Actually, in the case of a retrovirus, the AIDS virus, which is R-E-T-R-O, retrovirus, what does this mean? Viruses in general are thought to be the smallest replicating organisms that require other cells to grow themselves in. Viruses are not capable of reproducing themselves on their own outside of living tissue, is the dogma of the scientists today. Viruses must inhabit another cell for eventual growth and reproduction. Bacteria, fungi, and some other organisms actually are capable of growing outside of tissue. In other words, they don't have to inhibit or inhabit other tissue to reproduce themselves. They can grow on tissue culture plates, such as bacteria. But viruses must grow inside of tissue, which requires that there be living human or animal tissue for them to replicate in. Now, if we look at the word retrovirus, we know that this is a small self-replicating organism which grows inside of living tissue. Now, what does the term retro stand for? The term retro, in the case of this virus, stands for the fact that contained within the AIDS virus and other so-called human retroviruses or other animal retroviruses are small enzymes known as reverse transcriptase. That is where the word retro comes from. The reverse transcriptase, which is the RE comes from reverse, and the TRO comes from transcriptase. That is an enzyme in the AIDS virus which actually is responsible for duplication of the genes of the AIDS virus, which are in an RNA form, different than human form. Human genetic material is in a DNA form. So if the AIDS virus is to insert itself into human material, somehow after infection of the cell, what happens is this enzyme duplicates the RNA of the AIDS virus into a DNA form and actually inserts that into the human DNA. If you have an example, here's a cell, and inside this cell, here's the human DNA. What happens is the AIDS virus genes get in and are actually duplicated into DNA form, copied by the reverse transcriptase. That information is then inserted into the genetic makeup of the human cell. This is now an AIDS virus residing in the human genes, which then sends out a signal for production of a new AIDS virus. So, the RNA is the genetic information of all retroviruses. It's copied into the DNA form by the reverse transcriptase, inserted into the genes, and subsequent production of new virus.
To better understand this subject and to help us appreciate the importance of Dr. Strecker's work, I'm going to briefly clarify and emphasize some of the information in this presentation. Virology is the study of viruses. It deals with tiny living organisms visible only with the use of the most powerful electron microscopes. In fact, hundreds of thousands of AIDS viruses can easily fit on the head of a pin. What makes the AIDS virus particularly deadly is its ability to not only invade and neutralize human cells, but the virus's ability to put its own genetic material inside the human cell's genetic structure, thereby allowing the virus to use the human cell as a kind of virus factory, reproducing more viruses from the human cell's raw materials. Unlike larger organisms like bacteria, viruses do not respond to conventional medical treatment, much like the common cold virus cannot be treated effectively by drugs.
Now, how does this AIDS virus, which is a human retrovirus of the D type, how does this virus affect humans? Basically, the immune system of humans is broken down into two parts. It's very simple. One is called B cells and one is called T cells. B cells are derived from the bursa fabricius, but the easy way to remember B is that they control basically production of antibodies and control bacterial infections, therefore B rather than bursa. You can think of it as bacteria. T cell systems control opportunistic infections such as pneumocystis carinii pneumonia, the production of cancers like Kaposi sarcoma, and other microorganisms such as tuberculosis. So that if you wiped out the T cell system, you could see the arisal of opportunistic infections such as pneumocystic carinii pneumonia, a hallmark of AIDS, Kaposi sarcoma, an alleged sarcoma, and another hallmark of AIDS, or other diseases. If you wiped out the B system, you would have trouble protecting yourself against bacteria and perhaps in developing antibodies. Now, in the case of the AIDS virus, once it infects the human body, selectively destroys the T4 cells of the human body. The T4 cells are a division of the T lymphocytic system, the one that helps us control cancers, fungi, pneumocystis carinii pneumonia, and other organisms. The AIDS virus selectively leads to the destruction of those cells. If you look at the overall incidence of these new so-called human retroviruses, you'll discover the following. First, there is HTLV1, a new human retrovirus which is responsible for T-cell leukemia. There is HTLV2, which has this appearance, which is responsible for the development of hairy cell leukemia. And we have HTLV3, which looks like this. These are the best known so far, which is the AIDS virus, responsible for AIDS. Now, interestingly enough, when you put these viruses into tissue culture, what happens? This virus is proliferative in tissue culture. It makes things grow. It's not surprising, therefore, that you might see the arisal of a T-cell leukemia because the virus's very nature is to make the cells proliferate. This virus is proliferative in tissue culture, results in humans in the development of hairy cell leukemia. And again, by its basic proliferative nature in tissue culture, it's not surprising that you can see arising hairy cell leukemia. This virus in tissue culture is destructive. It was one of the reasons that they had trouble getting enough of the virus because suppose this was a tissue culture, every time they put in the AIDS virus, what would happen was they'd come back in a few days and the tissue culture would be dead. Basically, there would be left just debris with very little virus, no living cells. That is basically what happens in human beings, because in humans, we are nothing more than walking tissue cultures. So, suppose we have a human being here. He gets infected with the AIDS virus, and what happens? Eventually, the AIDS virus wipes out his T4 lymphocytes, destroys his thymus most probably, and as a result, relieves him immune-suppressed, immune-compromised, and susceptible to the development of infections such as pneumocystis carinii pneumonia or the arisal of cancer such as Kaposi sarcoma. These have been the basic hallmarks of the AIDS virus. But again, let's look at the overall picture. And the overall picture is that suddenly in humans, we have an explosion of disease, not just AIDS, but other retroviruses, HTLV1, a T-cell leukemia virus, HTLV2, HTLV3, HTLV4, which is a new, recently recognized AIDS virus, also known as HIV2, HTLV5, which is the cause of mycosis fungoides, and HTLV1 lookalike, which looks like this. I call it 1 lookalike. Now, even looking at, say, let's throw AIDS out for a minute, you have to ask yourself, where did all these other viruses come from? And my explanation as to where these other things have come from, along with the AIDS virus, is the following. If you look at animals, particularly cattle and sheep, you'll discover an interesting phenomenon. In cattle, there's a virus known as bovine leukemia virus, which has the exact same morphology, which means shape, the exact same relative molecular weight, the same magnesium dependency, and the ability to produce B and T cell leukemia of cattle, and is proliferative in tissue culture. If you look at bovine syncytial virus, you'll find another virus of cattle, which has the same shape, the same magnesium dependency, the same basic appearance, and produces hairy cell leukemia in cattle. If you look back, you can discover a virus known as bovine Visna virus, which has the same appearance as AIDS, the same molecular weight, the same magnesium dependency, and in 1974, either 1 or 3 here, bovine leukemia or bovine Visna virus, was producing pneumocystis carinii pneumonia in chimpanzees. And if that isn't AIDS, I don't know what it is. Now we have HTLV4, which may represent a recombination between Visna and HTLV2, or bovine syncytial virus here, which is a new AIDS virus, growing, just identified, which has this appearance. We have HTLV5, which I'm not sure in animals what it is, and we have HTLV1 lookalike.
You'll recall that Dr. Strecker has shown us that the AIDS virus selects and destroys T4 cells. These are the cells in our bodies that protect us against the development of cancers. Now, persons infected with the AIDS virus have T4 cell destruction and subsequent development of specific types of cancer. These include Kaposi sarcoma and pneumocystic carinii pneumonia, which are fatal. Looking at the overall virus picture in a general way, Dr. Strecker has pointed out that there are several other deadly retroviruses besides the actual AIDS virus, which are infecting humans and causing cancers, including cancer of the blood, leukemia. Dr. Strecker makes an interesting and startling correlation. These human cancer-causing retroviruses, including AIDS, all have striking similarities to animal viruses, but not from the green monkey, from cattle and sheep, known as bovine and Visna viruses. Now, the implications of these similarities between human and animal viruses is disturbing. How did animal viruses get into humans? As we shall see, this subject begins to reveal the true origins of the AIDS virus.
Now, why do I bring up the question of cattle viruses and sheep viruses when everybody says that these viruses came from monkeys? I say that for the following reason. If you look at the genes of the AIDS virus, the genes of the AIDS virus don't look like monkeys. The genes of the AIDS virus, in every paper published to date, look like the following: bovine leukemia virus of cattle or Visna virus of sheep. Now, these are retroviruses of animals, and these viruses are known to cause brain rot in sheep and to cause leukemia of cattle. So, is it possible, I mean, it was my question, is it possible to cross these two viruses and make AIDS? Now, of course, if you go down and ask your local AIDS expert, is that possible? Most of them will probably lie and tell you, no, that's not possible, that's just nonsense. But the truth of the matter is that AIDS viruses, in a sense, are much like humans, in that if you have bovine leukemia virus on one hand and Visna virus on the other hand, and you simultaneously infect a human tissue culture, what comes out of that infection is not only the original parents, bovine leukemia virus and Visna virus, but what comes out of that is every possible recombinant that will grow. In fact, not only that, also comes out of the ones that won't grow, which you can't identify because some of them down here are recombinants that come out, but they won't reproduce. They're called, in fact, in retrovirology, those are called incompetent. They're not capable of reproduction. You might call them impotent. They can't reproduce. So we get out not only the competent, but the impotent. So, in fact, these viruses do, in a sense, not only reproduce themselves, but they make babies, which are different than the parental strains. It's just like each of us. Each of us is a recombination of our fathers and our mothers. If you say, well, we look at the AIDS virus, and we say, well, it's only 50% identical to Visna, and it's only 50% identical to bovine leukemia. And they say that's, quote, distally related. It's like saying I'm distally related to my mother or my father because I only contain 50% of their genes. Of course, that's nonsense. The fact is that containing 50% of the genes of the virus, you could be the direct descendant of the parents. It's the same as each of us. Each of us is a direct descendant of our parents, and we contain 50% of our mother's genes and 50% of our father's genes.